Já estamos online, Marcelo, pode começar.
Good morning, everybody. I would like to start our morning section, November 24, BSB 2020. Gonna have 5 presentations, and I would like to start the first presentation from Elvismary, Molina das Armas, Maristela, Holanda, Daniel de Oliveira, Nalvo, Almeida, and Sérgio Lifschitz. Evaluation of Bruijn graph algorithm for the Novo fragment assembly methods. I think Dr. Elvismary gonna present it, and please, you can start your talk.
Okay, good morning. I will use a pre-recorded version. Let's share the screen. Good morning, my name is Elvismary Molina. I'm associated with Lab Bio PD at Pinheiro, leading by our professor Sérgio Lifschitz. And today I will present our namely a classification of the Bruijn graph approaches for the Novo fragment assembly in collaboration with Professor Maristela from UNB Brasilia, Daniel de Oliveira from UFF and generation sequence dramatically lower the cost for sequencing nucleotide and increase it through by order of magnitude. However, they are not be able to obtain a complete strain of DNA. Instead, they generate a small fragment called reads. Since the reads are obtained by curing at random position and there may be sequencing errors, this technology tries to read each position of the genome several times, which is defined by coverage, generating a large number of reads with a high degree of redundancy. To obtain representation of complete genome from these reads, computational methods have been used in more complex where there are no reference genome, which is known as the Novo assembly. In that case, have been widely used the Bruijn graph data structure. In terms of computational complexity, the assembly might require high-performance computing platforms for large genomes and the processing of large volumes of data because of the high memory requirement of the Bruijn graph. Thus, the objective of this work is to give an overview of the main strategies applied to minimize the memory requirement for the Bruijn graph from the Novo fragment assembly. For this, I will present the elements that may impact the memory requirements for the construction of the Bruijn graph, and also I will talking about the main classification of those approaches, giving more detail about some data structure optimization, for example, data structure based on hash and block filters, and external memory processing strategies. The Bruijn graph could be defined through K-mers. A K-mer is a substring over a read with a specific key length. Consecutively, K-mers share a K-1 elements. The Bruijn graph represents overlap between K-mer in which the set of vertices B is defined by a set of unique K-mers over a given set of reads, and a set of X E contains the X in which X is defined by a set suffix-prefix overlap of length K-1 between two K-mers that occur in at least one read. K-value plays a main role in the Bruijn graph. It determines the number of elements of the graph. Also, I define the shortest length for overlap between two reads, and depending on it, the algorithms over the Bruijn graph will be able to distinguish between native repetition of the genome and a repetition given by the sequencing errors or high coverage. Because of this, biologists usually run the assembly with several key values and select the best results. For the graph construction, all K-mers are stretched from the set of n reads with length m. It is necessary to identify all the different K-mers to set of nodes, which is multiplicities and the x, through a sorting or lookup over data structure. This process is characterized by a huge number of elements to process and a high level of redundancy. Computational requirement for the Bruijn graph construction depends on K-mer processing and a particular key. The number of K-mers significantly affects the execution time, while the number of unique K-mers significantly impact the amount requirement of main memory. Assembler software based on the Bruijn graph implements the whole process to construct the graph and also to traverse this for content generation. Some examples are Path, Abyss, Novo, and Velvet. On the other hand, there are a set of software called K-mer counter responsible for generating histograms about K-mer, so they only get involved with the K-mer generation, identification of unique K-mer, and counting. So there are several techniques to reduce the memory footprint for assembling process. We have classified them into two general groups. The first group considers solutions that reduce the amount of data as a cause of non-exact representation of the Bruijn graph. The goal is to reduce the data to process as much as possible by removing or sampling until it is possible to execute the assembly with available resource. However, it may affect the final quality of assembly. In that group, we also include those approaches that use probabilistic data structures as Bloom filters. The second group considers solutions that increase the memory resource for the same amount of data for an exact representation of the Bruijn graph, extending the computational resource through parallelization, for example, and through some techniques like partitioning or distributing in a certain memory. Regardless of the approach used, some data structures optimization are developed to store the set of vertices and eggs based on, for example, hash tables, probabilistic data structures like Bloom filters, tree vectors, and so on. Hash tables have been widely used to represent the Bruijn graph as data structure to store the K-mers. A hash table can map keys to values using a hash function. Its primary advantage is that lookup, insertion, and deletion operations run in constant time on average. The problem is when the hash function points to the same bucket for two different elements, so the collision must be handled. In that case, it could be necessary the use of another data structure into the bucket to improve the lookup time. Once the bucket, the K-mer itself could be stored using a specific codification. 2 bits per base is commonly used. Therefore, the total amount of memory needed to store the Bruijn graph depends on the key value, the number of K-mers stored in the table, and the needed of optional data structure for collisional handling. Some examples of the application that use hash tables to store K-mers are, for example, Babel disassembler, which uses 2-bit per base codification for K-mers, and a split tree to handle collision into the buckets. Also, Abyss uses a 2-bit per base representation and a sparse hash table with a linear proving scheme for storing K-mers. In the case of jellyfish, it is a K-mer counter that uses a vegetative function for hash and a core part of the K-mer in the index. And finally, Miraculous is an assembler that uses a novel lightweight hash with multiple hash functions to avoid explicitly storing the key themselves. In the case of not a star representation of the Bruijn graph, we have the approach that uses probabilistic data structures like Bloom filters. A Bloom filter is a space-efficient data structure for representing a given subset of elements T from the universe U. A Bloom filter consists of a bitmap P of size M and a set of P distinct hash functions. Initially, all bits of P are set in 0. An insertion of an element is done by setting the bits corresponding to each hash function to 1. This approach has the disadvantage of having false positive nodes as a direct consequence of false positive in the Bloom filters, which can create false connections in the graph. Recent works are focused on how to minimize the set of false positives using some structures. In the table 1, it is possible to see some approach focuses on reduce and store the set of false positives in Bloom filters. For example, missing counter is an algorithm that two steps to correct false positive and use one by counter per K-mer and cascading Bloom filter in which they artificially apply a Bloom filter to represent the set of false positives. In the side of a exact representation of the Bruijn graph, we have some approaches like external memory sorting, partition processing of the Bruijn graph in relational database manager system, and a final approach focuses on external memory processing with the reduction of elements to externalize. In general, the approach that implements partition processing generates the collection of all K-mer from reads, distribute them using a distribution function. In some cases, the same K-mer is copied to more than one disk partition. Then they process the partition in main memory, identifying the unique K-mers, and after that, they merge the results of all partitions in this. The total size of the memory need in this is the sum of the partition size, and the maximum size to be used as a main memory is not previously known and depends on the large partition and the partition function used. The total number of I/O operations depends on the number of K-mer distributed over disk and the size of the partition. The K-mers are copied on read several times from the disk, generating a greater amount of input and output operation. As another case of external memory processing is an implementation that our research group developed, implementing the K-mer mapping into a relational database manager system based on Velvet algorithm in order to mitigate the high memory requirements by using persistent structure in external memory. The objective of the research was to evaluate the impact of different indexes for K-mer, like bit+3 index using a K-mer string as primary key, also a hash index of a K-mer string, and finally the hash index of a K-mer pin minimum substring. As a final example of external memory processing, I will comment about another solution that our group proposed for external memory processing with a reduction of elements to externalize, in which the Bruijn graph is generated through iterative reduction steps using extra-compact Bruijn graph, in which the nodes are structures called the K-mer for finally obtaining the same Bruijn graph as other approach. As a conclusions, we have the most critical parameter in the Bruijn graph is the key value, which impacts the assembly accuracy, the number of vertices and eggs, and the memory requirements. Several techniques have been proposed to construct the Bruijn graph. This paper classified those approaches into two main categories, one for an exact and complete graph, and another for non-exact Bruijn graph representation. Considering the solution based on probabilistic data structures used for an approximated Bruijn graph representation, the most explored data structure are the Bloom filters. This data structure allows vertices to be stored independently of their number, but at the cost of false positive. Finally, we could find some solution using external memory to construct a graph with an exact representation. They process all K-mers in external memory. A new algorithm building an accurate representation without the need of process all K-mer have also been found. In conclusion, this paper presents in details the main theoretical and practical aspects related to the Novo assembly, particularly the Bruijn graph structure. We have enumerated certain approaches to reducing the memory requirements for the Bruijn graph construction. We have proposed a comparative view of a citing solution in literature.
So, okay, this is a presentation, and I would like to hear any questions. I am trying to, okay, start video.
Thank you, Dr. Elvismary. Do you have any questions from audience?
Yes, I would like to talk. So, congrats on your work, Emma. Works like yours are very important to establish the state of the art, particularly for the assemblers, which memory use details are often hidden from our eyes. And my question is, maybe I couldn't see this in a presentation, or maybe you did not talk about, but could you verify a K-mer size that is better or worse depending on the memory strategy? I mean, depending on the memory, the strategy of memory, and the size of K-mer, could you identify a strategy which is better, for example, when the K-mer is bigger or smaller?
Okay. Yes, in some papers, they expose the final K-mer size. It is important to know that each paper, each research presents results based on a few tested, a few K-values, and few 1 or 2 or 3 databases. So they generalize the results, but certainly the results depend on the K value and the data set. So in Bloom filters, we know a few smaller size for K-mers, for example, from the development of the research through many papers. They reach from 30, 50 bits per K-mer until 8 and 9 bits per K-mer. But this is approach that is not an exact representation of the Bruijn graph, and it is possible to occur false positive, and could be a solution, but it is not exact. Some future work need to evaluate how much viable are those solutions in practical and real data sets and real studies.
I see. Thank you.
Thank you. Do you have another question? I have a question, Dr. Elvismary. Have you compared the efficiency of your proposal, of algorithm proposal, with some other available?
Do you mean if you will compare the algorithms to
Yes.
generate the content, the final the final assembly? No, our approach is based on data structure to support the Bruijn graph, and how are improve the size of the amount of memory needed. But our work doesn't compare the algorithms itself to generate the context, the final context.
Marcelo, Emma, do you know anyone else who is working in the same approach that you, using a database to serialize the K-mers or something like that?
Okay.
It seems to me that you are the only one who is working on it, and what is a very good thing, because for example, for some places that we have not so efficient computers and resource, it could do work very well. That if you think that we can assemble genomes using computers with a few memory, but I not recall seeing anyone else working on it. Have you find some competitor for saying?
Okay, about the processing and generate the Bruijn graph, my PhD thesis was about an algorithm to construct the Bruijn graph. This is the first phase to the whole process, and it is based on an approach to externalize K-mer process, but pre-processing the K-mer, reducing the amount of data to externalize. That is a mix between the algorithm and data structure. Also, we will have a presentation of Julio that he's working in the phase to process the Bruijn graph. This is the part of process, and in the last year in this event, I was presented at work about K-mer classification by using machine learning techniques. That is because the K-mer processing is not only interest to assembly. It is the process interesting for other studies, genomics, proteomics, and so on.
I see. But still, these works are from your group, right? So it seems to me that you and your colleagues are pioneers in this area or of exploring database as a alternative for low memory computers in genomic assembly. Am I correct?
Maybe, yes.
Congratulations on it.
Thank you. The researchers are focusing in improve data structure in main memory because it is quickly. Well, we are trying to. We assume that there is at any time the available main memory will not enough to process all the reads and information that we have resulting from sequencing techniques. And so try to improve the processing using techniques in second memory or external memory.
Very good. Thank you again.
Okay, thank you, Elvismary, for the nice presentation. I think we can move on to the next talk. Will be held by Marcos Braga, Kenny Pinheiro, Fabrício Araújo, Fábio Miranda, Artur Silva, and Rommel Ramos. Redundancy treatment of NGS contigs in microbial genome finishing with hashing-based approach. I think Dr. Marcos Braga will hold the talk.
Good morning.
Can start.
Good morning. Can you hear me?
Good morning. Can
Morning. Yes.
Yes, you can hear me.
I will project my screen. Can I start?
Yes, can move on. Yes, can see you.
Thank you. Good morning. My name is Marcos Braga, and on behalf of my colleagues, I will present now the study, Redundancy Treatment of NGS contigs in Microbial Genome Finishing with Hashing-Based Approach. That was accepted as a full paper.
Marcos, just a moment. We are not seeing your screen. It's not sharing.
Okay. Sorry. I commit this mistake. I think. What about now?
That's okay now.
Thank you. So this work was accepted as a full paper at the BSB 2020, and I will present you now. First, I'm going to make some initial considerations to contextualize the theme. Repetitions have always been technical challenges for sequence mapping and assembly tools. NGS sequencing with short reads and high throughput made these challenges even more difficult. From a computational perspective, repetitions create ambiguities in alignment and assembly, which can lead to distortions and errors in the interpretation of results. Most assemblers have addressed this problem, in particular overlay-based and the Bruijn-based methods. Both create graphs of different types from the sequencing data. Repetitions can cause ramifications in these graphs, and the assemblers must guess which branch to follow. And incorrect decisions create false associations, generate chimeric contigs, and wrong copy numbers. Also, it's common for an assembler to create a chimera by joining two distant regions in the genome, and in the end, the reads eventually align with the wrongly assembled genome. So, reads coming from different copies of the repetitive region on genome may be wrongly assembled as a unique contig, also called a repetitive config. Specific errors inherent in sequencing platforms also affect the quality of the generated assembly. This leads to consider to use different assembly tools, giving preference to those that have greater tolerance to errors observed in the platform of interest. Hybrid assembly approaches use different sequence platforms data, or different fragment sizes, or even data from distinct assemblers. When assembling with different assemblers and different methods, the resulting contigs are merged, generating a much larger amount of them. These contigs, in general, represent different regions of the genome and lead to different gaps. The large number of contigs produced requires considerable computational and human resources for analysis, identification of assembly errors, and elimination of bases with higher probability of contiguous edge error, which prevents the extent of overlap due to mismatch errors. This is responsible for significant increasing in the amount of generated contigs and therefore subsequent redundancy on data. And so redundant contigs difficult so much the genome finish process. Well, this work presents a hybrid computational method to detect and eliminate redundant contigs from microbial genome assemblies. Here we can see a brief overview of the methodology. Today's experiment, we used the dataset called GAGEB or GAGE-B. It is very commonly used to assembler competitions and to evaluate assembler performances. It consists of eight bacteria divided in 12 sets sequenced on Illumina HiSeq and MiSeq. On the right table, you can see the 12 species, their genome size, their GC content, and which technology it was sequenced, HiSeq or MiSeq. Well, in this experiment, we performed a hybrid de novo assemblies with two assemblers. One is the Spades, that is a de Bruijn graph assembler tool, and Fermi, that is a OLC assembler tool, overlap layout consensus. To perform the assembly with Spades, we use also the K-mer-based to improve estimating the K value, the best K value for the assembly. And at last, to evaluate and measure the performance of the assembly, we use the software Quast. Well, in the application, we present a computational method we call a BFLSH to detect and eliminate redundant NGS contigs generated by the Novo assemblers. The approach uses two hashing-based techniques. The first is a Bloom filter. The Bloom filter performs the exact string match to detect the duplicated contigs. And then we use in the second unit a technique called Locality-Sensitive hashing, or LSH. The LSH performs the approximate string match to similar contigs detection. The application was developed in Java and performed in this computer. We choose the similarity threshold of 75% to compare the similarity on contigs. In the first unit, the Bloom filter unit, we have the same behavior. First, to define, a Bloom filter is an array of bits, in this case, that represents a set of contigs of fixed size. A Bloom filter is used commonly to perform membership tests when I like to check if one element is member of a certain group of elements or a certain set. I can use a Bloom filter that is a very quickly and with low storage capacity technique and structure. The contig itself is not stored on the Bloom filter, but the indication whether it is present or not. Once Bloom filters are probabilistic structures, it is possible to affirm with high probability if one contig is already a member of the set. But once Bloom filters do not allow false negatives, it is possible to ensure if one contig does not belong to the set. I will describe briefly the behavior of the unit, the contig unit. We have two basic operations. The first is called the insert operation. So in the bottom, I have the Bloom filter itself. It's an array of bits. In this case, just for example, we have a Bloom filter with size equal to 20, from 0 to 19 positions. It's a bit array, so we can put zeros or ones. In this case, we initialize the Bloom filter with zeros. So we also have hash functions. In this case, it's a standard Bloom filter. So we use uniform hash functions. The uniform hash functions have the property to spread the codes between the space we have. The space we have here is from 0 to 19. Then the codes shall be generated in this range. We use three hash codes, three hash functions, MD5, CRC32, and SAJ1. So when we submit the contig1 to hash function, it generates three hash codes. For example, here we have 2 and 7 and 19, and this is the mapping for the structure. So these positions on the Bloom filter, 2, 7, and 19, shall be turned from 0 to 1. In a similar way, in the second hash function, we have three new hash codes, 3, 11, and 16, and those positions are turned from 0 to 1. And at last, in the third hash function, we have the hash codes 0, 8, and 15, and these positions are turned from 0 to 1. This is the insert operation. If other contig equal to contig1 is tested, the result is contig is already a member of the Bloom filter. So we can detect a duplicated contig, and the redundancy is also detected. The second operation on the Bloom filter is the test or query operation, when I want to test if one element is already a member or not of the set. The similar way is performed, but here a different contig generates different hash codes. And sometimes, in this case, is the case, the hash functions map to a position with 0. And as a property, the Bloom filter cannot allow false negatives. If just one position with 0 was mapped, I can affirm, I can ensure that this contig is not in the set. So if any hash code for contig2 mapped to a position with 0, contig2 is not a member of the Bloom filter. This is why, this is how we can detect with Bloomfield the duplicated contigs. In the second unit, we have the LSH, the locality-sensitive hashing stage. The LSH is divided into 3 steps. The first is the shingling, where contigs are divided into smaller sequence of size K, also called K shingles or K grams. The second step is the min hashing, where smaller sequences are converted into small numeric values or signatures without losing their original similarities. And the third step is the LSH itself, where a similarity of the pairs or candidates of signatures is verified through Jacquard index or Jacquard similarity or Jacquard distance. Here we have an overview of the LSH step unit. We have 2 contigs that shall be tested if are similar or not. In the first step, shingling, we generate the K grams, small sequences, many similar to the K-mers. These K grams for each contig is submitted to the second step, that is the min hash. On the min hashing, we will calculate the signature for each contig. And at last, the LSH step will say if these contigs, we verify if these contigs are similar or not. We'll briefly resume now. In the shingling step, we have here, for example, these 2 contigs, contig 1 and contig 2, and we generate the K grams. The K grams are generated in the similar way we generate the K-mers. They are just like the K-mers, but with small sizes. Here we have K equal to 3. So here we have the K grams for the contig 1 and the K grams for the contig 2. This is the shingling step. These K grams for the 2 contigs are submitted to the next step called the min hash. On the min hash, each K gram is submitted to hash functions. So we generate this matrix that's called the signature matrix. And in the end, we look for the lowest or the minor values in each column for compose the signature of the contig. This is why it is called min hash. We catch the minimum value in each column. So here we have as the minimum value 5, 1, 2, and 15. So this is the numbers or the values for the signature of contig 1. The similar way for contig 2, we have 5, 1, 6, and 2 as the minimum values, forming the signature of contig C2, 5, 1, 6, 2. And here the last step is the LSH itself. We will evaluate the similarity of the contigs through the Jacquard index using the Jacquard distance. And the Jacquard similarity is works very well with the min hashing functions family. So the signature similarity is the number of the min hash functions that match. It's simple. Here we have 4 values, and here 4 values. And we have 5, 1, 2, 16, 5, 1, 6, 2. We have 3 in 4 values that match. So we have a similarity of 75% or 0.75 or 3 in 4. As we define 75% as a threshold for similarity, we can say that these 2 contigs are similar. This is the way we can perform the approximate string match in the application. So to perform the benchmark and evaluate the performance of proposed model, 3 other methods were developed or used. First, the AGS-BLAST-N that is based on the Boros-Wheeler transform to perform the alignment, the simplifier, and the great celebrity, the CD hit. The comparison between those methods and BF-LSH is shown for spades and Fermi data. First, we show here the percent reduction for also for spades data, and then for Fermi data. Then we have here in bold the 2 values of best performance of reduction of redundant contigs, okay, in spades data, and here for Fermi data. The reason for Fermi data have many high percent in reduction is that the Fermi assembly was not that good as the spades was. So comparing with the 3 other algorithms, we have here the number of contigs in the raw data produced for the assemblies, in the assemblies, and we have the reduction performed, the number of contigs after the reduction performed with BF-LSH, AGS-BLAST-N, simplifier, and CD hit. For spades data, CD hit achieved the best results for 10 in the 12 species, but BF-LSH achieved the best results for 2 species and achieved the second best performance in reduction the redundancy for the other 10 species. To Fermi data, the CD hit achieved the best results for all the 12 species, but also BF-LSH achieved the second best results for 12 species. And as a conclusion, Bruijn filter and locality-sensitive hashing techniques can effectively be combined to find similar items in biological sequences, especially in the genome finishing task. The probabilistic nature of the hash functions makes possible for errors to occur, false negatives and false positives, but these can be minimized with appropriate handling. A trade-off must be achieved between the Bruijn filter's main parameters, such as the number of hash functions, the filter size, and the number of elements in the set. Thank you. I'd like to make some acknowledgements. Above all, I have to thank God. I have to thank my institution of work, Universidade Federal Rural da Amazônia, my institution of study, Universidade Federal do Pará, and also thanks to Rede de Ciências Ômicas, Computational Biology Research Group, my research group, Núcleo de Pesquisas em Computação Aplicada, my doctoral program, PPGBM, Programa de Pós-Graduação em Biologia Molecular, and at least but not last, the CNPq that has funding this research. Thank you so much. I appreciate it.
Thank you, Dr. Marcos, for a nice talk. We have some questions here. I can, Dr. Sérgio, you can make your question. Are you there?
Yes.
Just a moment. I will finish this presentation.
Oh, you didn't finish? Sorry. Oh, okay.
Oh.
Okay. Okay.
Thank you. Please.
Dr. Sérgio is there? Can...
Yes, Marcelo, I'm here. Just a quick question, Marcos. Congrats on your presentation. It was very nice.
Thanks so much.
Interesting work. And I have many questions, but a simple one is related to the previous question by Valdeir. He was asking about other works related to the size of the K for the K-mer. Can you talk about a bit more about this K-mer gene you've mentioned before?
The K-mer gene is a tool we commonly use to improve assembly when we have deep Bruijn graphs assembler tools. So we first submit the data, the raw data, to K-mer gene, and it estimates the basic K value. So after these estimates, when I will perform the assembly with spades, for example, I put the best already calculated K value in the spade before the assembly. But for some cases in our experiment, 2 assemblies we did not achieve a good value, a good estimated value with K-mer gene. So we used the suggestion of the spades itself. But it's a tool we use it to estimate K-mer before we perform the assembly with the Bruijn graphs assembly tools.
And just to complete my question, this is valid for spades only or for any assembler, the K-mer gene?
For K-mer gene evaluates raw data and estimates the best value for K. I think we can use in each deep Bruijn graph assembler tool. I have used it for spades, but I think it's general, in my opinion.
Okay, thank you.
Thank you so much.
Okay, now we have a question from Giovanna Mota. Are you there, Giovanna? I can.
Did she type it? Did she type it in the chat?
Yeah, I can read it. I did understand very well how in the signature matrix, the K-grams are separated into columns. How is this distribution done? Is it random?
The signature?
Yeah.
Well, first, the LSH. First, the LSH approach is a near neighbor approach for hash functions. We have, for example, the behavior of a conventional hash function is spread the hash codes over all the space uniformly. In the LSH, the hash codes map to close space on the structure. This is how this kind of approach maintain the similarity of the data. This is a commonly used approach to perform approximate string match in hashing functions. But the mean hashing, the LSH is turned in 3 steps, and the mean hashing performs the same steps. First, for each K-gram, 4 hash codes, for example, 4 hash functions generate 4 hash codes, and I create that matrix. So for each column, I look for the lowest value to compose my signature. And those signatures are compared on the LSH stage to decide if they are similar or not. Well, this is the way it is performed. I don't know if I answered the question, but you can complement if you want. Thank you.
Okay, thank you. We have a question from Gustavo Tamasco.
Hello. First of all, thank you for your time. It was a really nice work. And I have a question for you about the contigs. I saw that your approach reduced the number of chimeric contigs, and I would like to know, like, if you have a way to measure this improvement, and because you are using the oh my god, I forgot the word. You are using the structure that increases the number of false positives, right? I forgot the name of the structure that you are using.
Bloom filter?
Sorry? Yeah, Bloom filters, yes. So you have a higher number of false positives. Can you measure how much this improvement is due to the factor of new false positives being injected to the analysis or not?
Can I complement? I have a very similar question. So I was thinking about it too, and my question is, did you check how the reads quality can impact these false positives or not?
Thank you. Okay. So first to Gustavo. We actually have a system that is mounted to perform membership test.
Mm-hmm.
So if one contig come and false positive are allowed, it means that if I check a certain contig 3,
mm-hmm.
and we have 1 for all the values mapped on the Bloom filter, I can affirm with high probability that that contig is on the set, but I cannot ensure because of the high of the false positive. So two probabilistic structures, just like such as the Bloom filter, we have not how to ensure that false positives will not affect, but we can minimize the effect of these false positives with handling the structure. And in the conclusion, I mentioned some techniques that we can use, changing the parameters, the number, the size of the Bloom filter, the number of hash functions. For example, if I use a lot much hash function, just like such as 5 or 10 hash functions, I will reduce dramatically the number of false positives. Anyway, I will increase the computational cost.
Yeah.
So this handling with parameters in Bloom filter can improve the performance and minimize the effect of the false positives. But we did not perform this evaluation of this impact. And to the second question, we did not evaluate also the impact or the effect of the reads quality in these false positives. But it's a further work to do. It's a good suggestion. I will take some notes about this.
Yeah, you compared your results with the result of the other approach, right? But of course, in this case, you use the same set of data, of the raw data, and it's okay. But maybe, and as you just said, for the future works, you could test different quality filters.
Yes.
And see how is impact
Yes.
on your reducing. If I allow it, I have one more question. Can I?
For me, there's no problem.
One more because we're out of time.
Oh, I will send you by email. Thank you. Good.
Oh, no problem. You're welcome. Thank you all. It's a pleasure to be here.
Okay, thank you. So thank you, Marcus. So we're going to move to the next presentation. A paper by Sérgio Lifschitz, Eduard Hermann Hausle, and Julio Osmar Pietro, Intensa, efficient out-of-core contig generation.
Marcelo, Julio is facing some internet problems, so I will share his video, okay?
Okay, go ahead.
Good morning. My name is Julio Omar. Today, I'm going to show you how we can use an external memory approach to generate contigs during the genome assembly process. Genome sequences is the process that determines the order of nucleotides inside a DNA molecule. The real genome is unknown. For that reason, modern instruments divide a genome into a set of many subsequences called reads. Here we have an example that are assembled into longer contiguous sequences called contigs. To create the contigs, two structures based on graph are used, the Bruijn graph and overlap consensus graph. This will work focus on the Bruijn graph. Suppose that we have a sequence. Here we have. We break the sequence into K-mers of length 3. For each unique K-mers, we have a node of the graph, and each between them are alignment between those nodes. To create the contigs from the Bruijn graph, we need to do some graph operations, such as graph contraction, and remove topological errors, such as tips and bubbles. The graph contraction is whenever a node has only one outgoing direct edge points out to another node that has only one incoming direct edge. These two nodes are merged. See the red cycle here. We have three unambiguous paths, and we merge them into one more graph, reduced graph. Tips are short ends branch of the main path. Possible causes of that include sequencing error toward one end of the read and low coverage. Again, in the red cycle, we see two possible tips, node A, I, and node F. We remove them of the main graph, and these are the results. Finally, bubbles are a divergence of a path into two branches after was joined together again into one path. Possible cause includes sequencing error toward the middle of the read. Here we have two possible paths between node A and H through E and B. And in this case, we remove the node E and keep the path ABH. But the approach using the Bruijn graph has a big problem, because in some cases, the graph is too big to fit in RAM. This has caused because two main issues. First, the organism's genome complexity. In this case, if, for example, the genome size is bacterial, if from bacterial, we can deal with that graph using a standard desktop and laptop system. However, in the case of human size, we need approximately 100 gigabytes of RAMs. And more complex organisms, for example, Y-spruce, we need more. The graph has more than 1 billion nodes, so we need more than 4 terabytes of RAM. And the second is the next sequencing platforms. They produce sequencing errors and genomic repeats. It can introduce new vertices and add each to the graph, significantly expanding its size and creating topological artifacts, for example, tips and bubbles. Consequently, if the size of the graph exceeds the amount of memory available, it will be necessary to increase the RAM. As in the near future, the size of the dataset will increase dramatically, and this situation will stress the different systems. There is a need for new approach to process all of this massive amounts of information in a scalable way. So, given the Bruijn graph, how do generate contigs when we do not have enough RAM to store it? We propose on this work to use an external memory approach to process the Bruijn graph. To increase the amount of graph does not guarantee that the graph will fit. To do with that, we break the graph in different intervals. That means all vertices are partitioned into intervals. The system only processes the related graph in the main memory on interval at times. The computation graph engine proceeds in different steps. For every step, it loads one of the partitions as available RAM. Then, it processes the vertices and each that belong to this partition and sends the partition back to disk. A different subset of partitions is then loaded and processed until all of them have been treated for each steps. This process is then repeated for the next step until there are no remaining vertices to visit. Here we can see an operation is measured as transferring from data between the disk and the memory. The external memory cost is will be applied to measure the performance of all algorithms. Here, I/O is a move the data between the memory and the disk. N is the numerous items in the problem instance, for example, the graph size. N is the numerous items that can fit into main memory, for example, the size of the partition. And B is the numerous items per disk blocks transfers. That means the numerous items we can transfer between disk and RAM. And here we have two main operators, scan and source. What the meaning of that? Or what we can use that? Because scan and source are optimal in the context of external memory cost. Contig generation algorithms. The first algorithms to contig generation is the contraction of non-branching path, as we mentioned. Here, for each node, we flip a coin into a value, tail or unhit. The vertices that flipped tails pick a neighborhood that flipped hits. Next, the vertices identified as tails with one outgoing edge send the required information to neighborhood. Next, the vertices, for example, C, marked as hits and tails and merge, and we recursively continue coalescing the graph until all unambiguous nodes have collapsed. Here, for example, the results of coalescing the graph A, the results are graph C. So what is the I/O cost for this? Here we have the edge set and scan the edge set multiplied for the loading of the longest non-ambiguous path, the longest non-branching path into the nodes. It's important to know that if the set of each is lesser than this measure, then for the edge is equivalent to scan the same set. So the I/O cost can be reduced only to scan the set of edge in a logarithmic path. Important, this result shows, and all the next results show how the approach to make an assembly using external memory is feasible. We can see how the main cost is from make and scan of the edge. Next, we have T removal algorithms. In this operation, the vertices having an edge, an in edge, for example, F, or an on edge, for example, I, are identified as potential tips, and a message is sent to the neighborhood. The vertices that receive, for example, C and D, that receive the message determine if they can eliminate the tips or not. Remove these tips, generate new linear path of the graph, and will be contracts. Here we sample how we eliminate F and E, and we only obtain the resulting graph. After that, we can contrast the graph as we made in the previous step. Here's the result. What is the cost? The cost of the graph is very optimal because the contraction only needs to full scan over all the partition. However, because after the eliminate the tips, we need contrast the graph. So the contraction step dominates the overall process. For this reason, this is the overall cost, as in the previous step. Finally, we have the bubbles. How we can do that? This approach of the bubble is based on breadth first search algorithms and has two steps, forward and backward. In forward, we identify the path, and in backward, we remove the select paths. To identify the path, every node stores an history. Here, example, where there is, what's the meaning of history? There is a path from node 8i to x through phi. So this means that the history, if the destiny node exists, we apply a heuristic to determine which path will be removed. In this example, for example, the node A is a beginning of the bubble. He sends the information to his neighbor. The neighbor stores the message, the history, and send another message, change the history. For example, in node C, we have, in graph C, we have node H have two histories, AB and AE. What's the meaning of that? Is that there is a path between A and H through B, and there is a path between A and H through E. After that, we identify all potential bubbles. So, how we remove this? In the backward step, just remember, we need to remove the redundant path and compact the graph. How in the node H, that we identify as the end of the bubble, we apply some heuristic depending on the bibliography. After that, we select which path will be removed. In this case, E, AEH. So we send a message telling to the next dot, telling to the graph one node will be deleted. After that, we remove the node A and we obtain the final graph. The cost of these algorithms is the same. Scan the edge and for the set. Multiply by the logarithm of B by logarithm of path by a factor of B. That means the longest length amount of bubble. What that means is the longest bubble in all the graph. What are the related works respect to deal with the graphs, the Bruijn graph size? First, we need some author make a sampling into the K-mer set. That means it performs a uniform K-mer sampling with a predefined sampling ratio. The problem with that approach is that, okay, we can reduce the graph, but we obtain approximate results because we are eliminate information in the K-mer set. Another approach is create optimized data structure, like what? Like way hash table, succinct data structure, and Bloom filters. This approach is very effective over the graph. However, these remain the same problems. If the graph size, of course, this data structure needs to increase. Difference, we are working, we are approach with the graph size is independent of the RAM. And finally, another approach is very straightforward. If the graph can fit into the one computer, so distribute the graph into many computers the way that all graphs fit in memory. As we can see, all these approaches are based on the principle that the graph need to fit into the main memory. This is different approach, but again, we only need a specific set of rank and we can process all the graph independent of the size with a fixed amount of rank. Our contribution, we propose of an off-of-course algorithms for dealing with contig generation using external memory. And we demonstrate that it becomes feasible to generate the Bruijn graph to obtain the mean contigs independent of the amount of RAM and independent on the size of the graph. And on Go works, we are implementing the external memory, the Novo genome assembling in contig generation using this library, STIPL. We need to evaluate the algorithm performance on real dataset, and we evaluate the performance on a complete assembly process. How a graph, how assembling can work using a fixed amount of ranks independent of the size of the graph. Thank you very much.
Thank you, Dr.
Thank you.
Thank you. It was a very nice work. Do we have any questions for Dr. Julio?
No, not yet.
Well, I have a question. Did you compare? It's a very nice proposal. But have you tested in real cases with other solutions? To other, did you evaluate your approach?
I don't understand if I evaluate my approach against
with real data comparing to.
No, right now I'm implementing this approach because it's difficult in the sense that the different library don't covers all the requirements for these problems. So we need to make some modification to, for example, remove the node, remove some each, the set on each. So it is a little difficult to implement that. But this is, we are working on that to implement in the next few months.
Okay.
It's important to know that I'm not working, I'm not talking about this is optimal. The main approach that is feasible to make an assembly graph using external memory. This is change in the evolution of the RAM memory and the data set in the Illumina set and genome database.
Doesn't it make slower down the process? If you use external memory, you don't get a slowdown.
That is the need to evaluate because right now there is more hardware, more SSD disks with high velocity. So if because of that, we need to implement another algorithms to deal with that. To make increase the performance using new hardwares, but much more cheap than a cluster or any other hardware.
Okay, very interesting. Do you have any question for Julio? Okay, thank you, Julio. We can move on to the next talk. Nós talks from Iago Blanco, Carlos Leonardo Araújo, and Adriana Folador. In silico pathogenomic analysis of Corynebacterium, pseudotuberculosis, biovar ovis.
Okay, do you listen to me?
Iago.
Yes, is it working? I will turn on my camera. 1 minute.
Okay.
Sorry, I didn't. Do you listen to me?
We can see you.
Perfect. Okay, I'm ready. Can I start?
Sure, can go on, can move on.
Yes. Okay, so good morning. My name is Iago Blanco. I am a biotechnologist graduated at the Federal University of Pará, where I have worked in the laboratory of genomics and bioinformatics, together with Dr. Carlos Leonardo Araújo and Professor Dr. Adriana Folador. Sorry. And today I will present my work entitled In Silico Pathogenomic Analysis of Corynebacterium subtuberculosis by Ovar ovis. Okay, to introduce that, we must consider that the bacterial genus Corynebacterium can have its species divided as pathogenic and non-pathogenic. Among the pathogenic or diphtheric ones, we can mention C. subtuberculosis, C. diphtheria, and C. ulcerans, which are listed and responsible for the transmission of casus lymphadenitis, ulcerative lymphangitis, and diphtheria. So responsible for the transmission of these diseases, some virulence factors and pathogenicity-related proteins should be considered. Considering Corynebacterium subtuberculosis, this species can be classified according to its ability to reduce nitrate in two groups. So nitrate reductase positive bacteria, they are classified in Biovar equi and can transmit ulcerative lymphangitis, attacking animals as horses and buffaloes, while those bacteria able to reduce nitrate are classified in Biovar ovis, which can transmit casus lymphadenitis, causing wounds and abscesses in the internal organs of small ruminants as sheep and goats, for example. So with the next generation sequencing technologies, it were observed great advances in the field of genomics, allowing the identification of revelations about pathogenicity mechanisms and process of pathogenicity in general. So in silico studies are those performed through computational analysis and predictions, employing bioinformatics and computational biology to make revelations about some organisms. Several processes relate with pathogenicity, as for example, adherence, iron uptake, secretion of toxins, invasion, and others in the host. So the application of comparative pathogenomics in this study allows the investigation of which genes are responsible for the pathogenic functions and how they work. Once it is possible to observe gene functions in silico and their respective conservation among different strains of the pathogen, with their respective functions related to pathogenicity, resistance, adaptation, and other aspects. So continuing, in order to identify the determinants for pathogenicity, some genetic factors should be pointed out, as for example, the pathogenicity islands, or PI, which are genomic regions with considerably low GC content and also potential pathogenic functions. So the analysis of composition of this loci, these regions, can reveal much about the pathogen's lifestyle, for example, and its infection mechanisms, as for example, conservation of virulence factors, VF. So those are known to be highly related with pathogenesis among different species and also strongly conserved among them. Several virulence factors are reported in the literature having roles in pathogenic processes. So besides that, also the presence of prophage DNA in bacterial genomes can be observed, and it's related with phage encoded virulence genes. What is possible to be observed and predicted through in silico tools. So in this study, we looked for the prediction of pathogenicity determinant for casus lymphadenitis in the genomes of C. subtuberculosis by Ovar ovis bacteria, which are able to transmit and cause this disease. Okay, talking about the methodology we use it, all of our analyses were made in silico. So firstly, the complete genomes of 33 strains of C. tuberculosis biovar ovis available at the time in the NCBI database were downloaded and also standardized to perform the further analysis. Initially, we obtained the genome files, standardized them, and generated the files corresponding to their amino acid and nucleotide sequences and functions of annotated proteins, so the known as PEP, NUNC, and function files. We used these files as inputs to perform a pangenomic analysis through the software PGAP to observe the orthology and conservation of genes among different strains. After that, we used the complete genome to predict virulence factors of these species using the VF Analyzer 2, if the virulence factor database. And further, the prediction of protein-protein interactions, what known as PPI, among these virulence factors using their amino acid sequences to observe high confidence interaction networks in silico. We also predicted the conservation of pathogenicity islands, PI, in the genomes and their respective compositions. The software Gypsy was used, which uses as inputs the query genome of bacteria and a non-pathogenic genome as a comparison, as a reference, and performs several alignments against different databases. For example, the NCBI BLAST and VFDB BLAST, which is the BLAST in the virulence factor database. So this tool provides composition, position of genes, GC contents, and also GC deviation of this specific loci, these regions. Further, we also compared different genomes of these species to analyze the synteny and conservation of pathogenicity-related genes. It were compared the genomes of tuberculosis 12C, FRC41, and 1002, generating images of conservation, translocations, and presence of specific genes through BLAST alignment. Finally, in order to predict the presence of phage DNA in the genome of tuberculosis 12C, we used the online tool FAST, which requires FAST files of the query genome, generating hits related to the profiled DNA through data alignment. Okay, so as starting with our results, through the in silico prediction, it was possible to find 4 major classes of virulence factors, organized in 12 types and 36 genes for the genus Corynebacterium. However, for our analyzed strains, only 20 of these 36 were conserved among all our analyzed strains in the study. The first class of virulence factor is the one related to adherence factors. It were found genes related to SPA-A and SPA-D pili. So pili, what's that? Pili are filamentary cell structures which provide adhesive and invasive functions to the pathogen. What is beneficial to the progress of infection in the point of view of the pathogen? It's reported that in Corynebacterium, the formation of pili by the sortase machinery. So, as well as in other pathogenic bacteria, not only for Corynebacterium. So the SPA type pili, this name stands for sortase-mediated pilus assembly pili. So these molecules, they help in the process of bacterial attachments to the host after infection. Some pili like SPA-A, B, and C are known to relate to the adhesion to human cells after infection, so helping the progression of the infection. Besides that, some surface-anchored pilus proteins are conserved also as virulence factors in this genus, of which only the gene SAPA was conserved here, as can be observed in the table. It is present in the genomes of most of the biovar ovis and in C. ulcerans too, other pathogen of this genus. Scientific literature suggests that horizontal transfer of these adherent genes in tuberculosis is a possible mechanism of transference of these virulence factors. So using the pangenomic comparison, it was possible to observe here that while some genes were conserved in all genomes, some of them were present in only some of them, indicating a role in process of pathogenicity, as will be discussed later. So continuing, the second class of virulence factor is the iron uptake factors found in this study that can be divided into ABC transporters, ABC type heme transporters, and iron uptake and siderophore biosynthesis factors. So the first one is known for its relation with regulation of virulence factors in different bacteria, like E. coli, Shigella dysentery, and Vibrio cholerae, for example. So the virulence-related cluster, FAG ABCD, which is observed in the table, is almost entirely conserved in almost all strains of this biovore, and it's also inserted in pi in pathogenicity islands, placed right next to phospholipase D, or PLD. That is a determinant factor for the caseous lymphadenitis. We can also note that the ABC type heme transporters, which encode ABC type heme proteins, are conserved in most of the biovore ovis genomes as virulence factors. For example, the HMA, HMU, T, U, and V. Also, the genes related to iron uptake, they can be found in this pathogenic bacteria once iron is known to be required for bacterial survival. So upon the infection process, bacteria tend to acquire host iron sources, like for example, the heme proteins and others. So the gene cluster CIU-ABCDE is known to be related with ABC type iron transport, biosynthesis, and siderophore production. This gene cluster has an iron-dependent regulation, and as exhibited to be highly conserved, it's suggested in the literature that has an essential function on the pathogenesis of these bacteria. So continuing to the next type of virulence factors observed, we identified some the class of regulation factors, in which we can highlight the diphtheria toxin repressor DTXR, which is a metal ion activated molecule that acts as transcriptional regulator in different species like Corynebacterium diphtheria, Jayceon efficiens, and also in all of our analyzed strains here. As the name suggests, we can associate to the diphtheria toxin repressor to the disease diphtheria, but we could find the conservation of this gene in all of our analyzed strains here. So the literature reports the presence of these factors in different species. So it's suggested to be acquired through horizontal gene transfer and having different roles and functions for different organisms. Continuing to the last part, we highlight toxin, PLD, or phospholipase D, which is the major virulence factor for caseous lymphadenitis, and is placed in a pathogenicity island, in a pi. So we could see it is present in all of our analyzed strains. It's known to cause host macrophage cell death, and it can be regulated by environmental factors, for example, temperature, molarity, and pH. So we can highlight the importance of these virulence factors, as most of them are conserved in almost all of our analyzed strains in this genomics study. So continuing, we also looked for the observation of pi in silico. We could observe that we could identify 11 pathogenicity islands with various genes annotated as virulence factors as expected, for example, as toxins, iron uptake proteins, and others. However, many of the components of these islands are still characterized as hypothetical proteins, which are coding sequences not annotated with the proper function or the proper product. So this highlights the need, the necessity for identification of these potentially pathogenicity-related molecules, once most of the components of pi are related to pathogenicity. So in figure 1, we highlight a synteny view of conservation of the main virulence factors identified here. Some of the most important virulence factors stand out, as for example, the FAG ABCD cluster and the toxin PLD, which are highly conserved in all compared strains and placed together. Among our main virulence factors, the operon HMU-TUV is also highly conserved with few differences about position, location in the 3 compared strains, as you can see. Besides that, the CIU-ABCD gene cluster is also highlighted with its importance in the process of pathogenicity, but exhibiting difference in position and orientation in the different genomes, so in different positions and frames of the DNA. So this is highlighted to the importance of different virulence factors. So besides that, we also observe that some other interesting molecules related to pathogenicity were found when analyzing the contents, the constitution of this pi in silico. So I highlight here below in the right part. So some molecules stand out, as for example, in the pi 5, the PTS sugar transporter PFOS, which is responsible for regulation of toxins in bacteria like Clostridium perfringens, and the proline immunopeptidase, known to be essential for virulence of Xanthomonas campestris, and also conserved in cystic tuberculosis. Besides that, in pi 7, it was possible to observe a non-annotated adenosine-related protein, which may be associated to the role of adherence in the pathogenesis of bacteria and the host. And in pi 8, we observe a sortase 1 and a collagen binding surface protein, which relates to, as mentioned before, the pathogenic related process of adherence. And in the pi 9, we could find another sortase 2 and some iron transport proteins as PHUC and FACT. Okay, so continuing, if we look for further details about the composition of pi, we could predict the composition, the pathogenic composition of these regions, this loci. So using the GPSI tool, we could observe that the complete genome of the reference strain, Corynebacterium subtuberculosis 12C, showed a total of 2,200 genes with a GC deviation of about 10%. So about 30% of these genes, or 683, were predicted to be related with pathogenic function by both employed approaches. We used the methodology of prediction of pathogenicity using the tool MP3, which resorts the approaches SVM, support vector machine, and HMM, hidden Markov model. So to predict proteins as pathogenic in silico, we used those which had an agreement between the two employments, SVM and HMM. So we performed similar analysis looking for the composition of the specific loci predicted as pi. So as reported in the scientific literature, these islands are known to have a genetic repertoire related to pathogenic processes. So most of these genes are related to pathogenicity, and also a lower GC content when compared to the rest of the genome. So this allows the prediction of these regions through in silico tools. That's what we did here. We could conclude that for all predicted regions, the calculated pathogenic percentages were higher than the complete genome when compared, and in general, they are formed by low GC content regions. We could find a mean value of 48.6% of proteins predicted as pathogenic in these regions, what is about 18.3% more than when compared this to the complete genome. And while the GC deviation of the complete genome was of about 10%, in this pathogenicity island, we calculated a mean value of 16.3%. So highlighting the importance of how to discover, how to predict these islands. They have some important particular characteristics. We also subjected the genome of Cypsidotuberculosis 12C to looking for prophage elements where we could observe the presence of 3 incomplete prophage regions. So one of them showed high similarity with other prophage regions found in other microorganisms described in the literature, like Mycobacteria, Burkholderia, and Synechococcus, for example. Most of the sequences found encode for functions related to enzymatic, structural, and ABC transport. But especially in the third prophage region found, predicted in this analysis, one protein stand out which allows and shows high homology to phage tail genes, which are reported to have various roles and work as adhesion molecules for bacterial attachments in the host upon the process of infection. So these tail molecules, they interact with the cell surface of the host and are subjected to strong selection pressure. So this is an interesting finding we got here. Continuing, we could see that most of virulence factors exhibit relation to well-known biological mechanisms. So we observed a predicted protein-protein interaction network, or PPI network, among main virulence factors of these species. It is illustrated in figure 2 here, so containing proteins of the cluster CIU ABCDE, HMU TUV, and FAG ABCDE, as described previously as important virulence factors of these genus. And we also observe a putative ABC transporter identified as AIG 0663.1, placed among them, showing some type of relation, but this is not a well annotated molecule in the available databases. So this is a computational prediction, but we can note through this methodology some experimentally proved interactions through data reported in the literature. For example, the interaction between CIU ABCD and FAG ABCD gene clusters. This was already previously proved experimentally, but our study is in agreement with this in silico and suggests an additional role of HMU TUV in the regulation of this pathogenic mechanism too. So the identification of associations between pathogenic proteins is directly related to their roles. That's what we are trying to find here. So our results showed a high conserved pathogenic network indicating relevance of virulence factors in this species working together. Well, as a conclusion of this work, we could investigate pathogenic determinants of Cypsidotuberculosis biovore ovis through in silico analysis. The comparative pathogenomics, the analysis revealed 20 main virulence factors conserved in our analyzed genomes, and these genes are essential for biological processes underlying the process of infection. Besides that, we showed the synteny of virulence factors, genomic lossy, and the presence and composition of pi in the genome of Cypsidotuberculosis 12C. So we observed that most of the virulence factors are found in these specific regions and organized in different gene clusters, which are described and known in the literature. We also found that this pi in general exhibited a higher potential of pathogenicity of its proteins, predicted to be 18.6% higher. And concerning the search for prophage elements, which are normally found to be related with pathogenicity, we could find 3 prophage regions: highlighting the presence of a protein with high similarity to phage tail genes, which is related to bacterial attachments to the host after the process of infection. Finally, we could also perform a protein-protein interaction network in silico, observing a high confidence interaction among main virulence factors of septic tuberculosis, highlighting the components of the clusters CIU ABCD, HMU TUV, and fake ABCD gene clusters, suggesting that combined roles are performed between them in pathogenic functions. So I thank you, all of you, for your attention. I thank the organization of the event, BSB, and also the Federal University of Pará, the CNPq, and ProPESP UFPA for structural and financial support, and the Center of Genomics and System Biology, where I have developed this study, and all of this team. Thank you. If you have questions.
Thank you, Iago. We have time for a few questions. Well, I have a question. Okay, you are Iago. You listen to me?
Yeah, yes, I am listening to you. I just can't close my presentation.
Okay, good.
Okay, now you don't see my screen anymore, right?
No, we are just seeing you. Yeah, okay, not the presentation, not your desktop. Iago, most of these pathogenic factors you have found, it's probably come from horizontal gene transfer, right? You mentioned this.
Yes.
And I was especially aware of this DTXR repressor, the diphtheria repressor.
Mm-hmm.
Look at this, but did you look up whether they must be a transfer from another bacteria, but they have a, have you look at the locus loss, whether there is some other genes together with the regulator, or whether the regulator regulates something? Do you have any idea the function of this regulator?
Well, if you are talking about the position of DTXR, I don't remember if exactly this is placed inside pathogenicity islands. But what I can tell you is that, interestingly, this gene is placed and it's conserved among most of the strains considered here in septic tuberculosis, not only for this biovir, but for the other equi, and also in Corynebacterium ulcerans. I have worked in a different research, and we found that. But we could predict that once it's important for regulation factors in process of pathogenicity in diphtheria, we can relate that somehow to influencing the pathogenicity in other species of Corynebacterium, as Corynebacterium tuberculosis. As I told you, this is conserved in other Corynebacterium too. Other besides the diphtheric ones, it's conserved too. So it's an interesting finding.
Yes, it's interesting. Yeah. Do we have more questions? Okay. If not, I would like to thank you, Iago. It's a very nice talk, for your nice talk, and we can move on. So to the next presentation. The next presentation is a short presentations from Renata Cavalcante, Miguel Ortega, Jorge de Souza, and Tetsu Sakamoto. Assessing the sex-related genomic composition difference use a K-mer-based approach, a case study in Arapaima gigas, o Pirarucu. Who is presenting this? Renata presenting this?
Yes, I'm here. Let me share my screen.
Mm-hmm.
Can you see my screen?
Yes, yes.
Okay. So, hi everyone, good morning. I'm Renata Cavalcante from Federal University of Rio Grande do Norte, and I'm going to present the short paper named The Sex-Related Genomic Composition Difference Use a K-mer-based Approach, a case of study in Arapaima gigas, Pirarucu. So, Arapaima gigas no Pirarucu is the largest freshwater-born fish in the world, and this species has a great potential for aquaculture because of the large size, fresh rate, and the color of the flesh. However, Pirarucu reproductive management in captivity is limited, essentially because of the difficulty in differentiating sexually this species, because of the lack of external dimorphism in these individuals. Previous studies on genome identify more specific regions suggesting the XY sex system for this species. However, the topic is still in debate because of the small size of these regions. Previous reports estimating 0.01 to 0.1% of the genome of this species as linked to the sexual determination. So, taking these points into consideration, our goals in this work were explore other genomic features in Arapaima gigas to find some clues about the genetic factors involved in the sex determination system, propose to access the genome composition of Arapaima gigas using a K-mer-based approach, and also identify region in excess or missing in one of the sex. So this is our pipeline, and we had as input 6 samples of Arapaima gigas, 3 males and 3 females, and the pre-processing step were used the cycle to trim the ribs, and the first step of K-mer analysis were count the K-mers. For this, you use the KMC to count the K-mers among the samples, and we choose a K-mer size as 23. After, we normalize these K-mers, and for this, we use the quantile normalization method, and we made this normalization with the in-house script in Perl. After, we filtering the repetitive regions because we saw the repetitive regions were most abundant in our samples. So we choose a repetitive regions from 1 to 8 per bit. And then finally, we count the K-mers with comparison the K-mer counts using a t-test or made in R. In a K-mer count, first we see the 6 samples and the reads average, and a result as a KMC or the total number of K-mer. So in this table, we can see the 6 samples, 3 males and 3 females, the reads average, a total number of K-mers, and the reference. So the female 25 and the male 25 were from a sequence for Duet-et-al, and the other samples were from another sequence for Vial-et-al. So we had 6 samples from 2 different sequences. And we can see the female 15 and the male 20 has a lower number of reads and there is a lower number of K-mers if compared with the other samples. And as I said previously, the K-mers most abundant were from repeated regions. As this difference between the number of reads and K-mers, we normalized the data using the quantile normalization method to make these K-mers, the samples, comparable among them. So previously in the A plot, we can see the female 15, female 20, male 50, and male 20, and seems to be a difference between the samples, a difference among the distribution of the samples. But after normalize the data, we can see in B plot, all the samples follow the same distribution. So after normalize the data, we could compare the samples, and in a K-mer count comparison, we saw the average comparison of the normalized K-mer count, and we saw the average for individuals of the opposite sex and the same sex. And in this table, we can see the repeat units with a significant difference, a low p-value, on the average of the normalized count between female and male. So we have in this table a repeating unit, the sequence samples, and the p-value. If we compare a group female against the group male, we can see some repeating units with a difference among these groups, between these groups. And as a AAGGC repeating unit highlighted in this table, we can see the females has a highest number if compared with the males. And we can see also in the repeating unit ACGTC, a female group has a highest number if compared with the males, and also in AAGC. And is there any repeat units as absent in alpha sex? So this table showed to us, this comparison using a t-test showed to us some repeats which were more abundant in one sex of the other. So we have noticed the excess of K-mers from repeated regions over or underrepresented in one of the sex indicate potential differences in the genetic composition between males and females of Arapaimagicas. The difference is not so express, and this corroborates with the previous reports that estimate 0.01 to 0.1% of the genome of Arapaimagicas linked to the sexual determination. And these repetitive regions have been reported in literature to have an important role on the sex determination in many vertebrates. As an example, we have a medaka, another species of fish, which has an XY sex system. There is a large stretch of repetitive regions on the Y-specific regions. So in conclusions, we can say that repetitive sequences found could be a component that could be used to determine sex in the individuals of Arapaimagicas. However, analyses with more samples to obtain a better statistical support are needed, because we have in this work only 6 samples of Arapaimagicas. And to summarize, a K-mer-based approach, a K-mer-based methods could be demonstrated to be an interesting approach to assist us unravel the sex determination system in Arapaimagicas. And in the future, we would like repeat this pipeline with another species of fish to see if we can find this difference among females and males also. To conclude, I would like to thank you all the funders that supported this work, and I also would like to thank you for watching my presentation. If you have any questions, I'll be happy to answer.
Thank you, Renata, for the nice talk. Do you have any questions for Renata?
I have.
Please, Valdir.
Hello, Renata.
Hello.
Thank you for the presentation. I was curious about the last thing you show. You say that there are other fish species that have some large regions that can be used to determine the sex, but this not occurs in the Arapaima gigas, right? But in your work, you have found some clues, some sequence that could be maybe used to determine. My curiosity is, have you tried to compare your findings to these regions on other species that already are known to be sex determining?
So we didn't compare our regions with the another species of fish yet, but we would like to validate experimentally these regions. We want to use these regions to make the fish experiments. So use these regions as species in another individuals of Arapaima because the major problem in my work were the sample, where we have only 6 samples of Arapaima. So in the future, in the near future, we would like to use these regions to try observe in more samples of Arapaima using the fish methods, but we didn't compare with another species yet.
Thank you.
Thank you.
Do you have any more questions? Oh, Renata, I have a question. In human, the Y chromosome is almost completely repetitive. There is few stretch of genes and mostly repetitive. I don't know in pieces, in fish, but you mentioned in your conclusion there is a lot of repetitive sequences also. How this would impact in your analysis? Because the K-mer should be getting this repetitive structure, and how this impact on your conclusion? You understand?
So I think I understand. So I didn't mention it in my presentation. The pirarucu, the Arapaima didn't has a chromosome, a sexual chromosome, if we see the cytologically. So all the chromosomes are seems to follow the same way. We can't see a difference in a cytologic. But our hypothesis is these sequences, these repetitive sequences as sites of heterozygosity could be associated with the sex-related system, because in the sites of heterozygosis is another part of my work, and we think those mechanisms. So in a individual, in a female individual, and as an example, has a on copy, single copy, and in the male has 2 copies of the gene. And we hypothesis is these dosage mechanisms is affecting the sex of these species, because fish is an interesting organism, because the temperature and the others mechanisms be associated with the sex determination. This is in this group. And I think I responded your question.
Yes. But it's a different system, right? You didn't mention, that's why I get confused. But that's good, nice work.
Yes, the previous work reports idea of XY sex system for Peru, but this topic is in debate because of the small size of these regions. Could be a XY sex system or could be another sex system in this species.
Okay, thank you.
Thank you.
Yeah, is there any more questions? So I'd like to thank you for the presentation. And I think there are no more questions. I'm gonna finish this section. I'm gonna thank all the speakers and the audience for being here. We can go now for lunchtime for the afternoon section. Okay, thank you everybody.
Thank you, Marcelo. Just remember that the afternoon session starts at 2 p.m. So I would like to ask two authors to arrive 30 minutes earlier to test the screen sharing and to test the sound sharing. So please, authors, come earlier to the afternoon session. Thank you very much.